Hi, and welcome back to educator.com. Today we're going to talk about homologous recombination and site-specific recombination of DNA. So this is linking back on the previous unit where we introduced the concept of homologous recombination, and now we're really going to get into the details. So as an overview, we're going to talk about all things homologous recombination. All right, so double holiday junctions, resolution versus the synthesis-dependent strand annealing, which we introduced last time. And then we're also going to talk about single-strand annealing, break-induced replication, meiotic recombination, as well as double-strand break repair in prokaryotes. And finally, we'll have a short overview on site-specific recombination. Now, homologous recombination, once again, remember, we can call this HR. Is genetic recombination in which the nucleotide sequences are exchanged between two similar, which are called homologous, or identical molecules of DNA. So two similar molecules of DNA are called homologous DNA. Okay, this will produce new combinations of DNA sequences during meiosis. Now, HR is often used in horizontal gene transfer to exchange genetic material between different strains and species of bacteria and viruses. That's what that is called. Horizontal gene transfer is the passing of DNA from one, let's say, bacteria to the other bacteria, as opposed to the passage through the new generations of proliferation. Okay, so homologous recombination that occurs during DNA repair tends to result in what is called a non-crossover product. So meaning it's fully repaired back to normal. It's as if nothing had happened. All right. Now we're also going to talk about forms of homologous recombination where we get what are called crossover products. So homologous recombination, reminders from last unit, it repairs double-strand breaks during S phase or G2 phase using long homologous sequences as templates for synthesis. So if we look here, the red and blue chromosomes are called homologs, okay, or homologous chromosomes. They are not the exact same sequence, but they are very, very similar. Okay. Now, the process of homologous recombination encounters several of these things that we're going to talk about. And generally, we start with resection. We then go to strand invasion, followed by holiday junction formation, and then either double-strand break repair or synthesis-dependent strand annealing. Okay. Now, after a double-strand break occurs, the mRNA complex will bind to DNA on either side of that break. Okay. We then have 5' to 3' resection, which allows us to have a 3' overhang. And then we have proteins coming in, binding these 3' overhangs, okay, specifically RPA and Rad51. We're talking about eukaryotic right now, okay? And this forms a nucleoprotein filament. What that means is it's a filament of nucleic acid, right, our DNA, and proteins, right, our RPA, our Rad51, and others. We then go through what's called homology searching. So we're looking for a piece of DNA that has roughly the same sequence. Okay, and then we go through a process called strand invasion. Okay, and that nucleoprotein filament invades the other homologous chromosome. Okay, so it goes into that DNA duplex. Now, if we're talking about being in mitosis, you'd be invading a sister chromatid. So you're using, those are identical chromosomes. That's the best time that you could do this. Or if we're in meiosis, you're using homologous chromosomes. So it's not the exact sequence, but basically the same. It's very similar. And when you go through strand invasion, you form what's called a displacement loop or a D loop. Now, DNA polymerase, once we've gone through strand invasion, DNA polymerase will extend the end of the invading 3' strand via normal DNA synthesis. And it's using the new DNA, okay, from that homologous chromosome as a template molecule, all right? And this whole strand invasion and initiation of synthesis will form a cross-shaped structure, which we call a holiday junction. Okay. Now, DNA synthesis will continue, okay, and that's continuing on the invading strand, so the original piece of DNA, not the homologous new piece, okay, effectively restoring the strand on the homologous chromosome that was displaced during strand invasion. Okay. The double-strand break repair is then completed by one of the two pathways. We either have the double-strand break repair, which we can also call the double holiday junction model, or SDSA, which is the synthesis-dependent strand annealing model. So let's talk about the first one, being the double holiday junction model. Okay. So the double-strand break repair pathway is unique, okay, in that the second 3' overhang, which is not involved in strand invasion, will also form a holiday junction with the homologous chromosome. Okay, so now we have what's called the double holiday junction. Okay, that will then get converted into recombination products by endonucleases. So we have cleavage events occurring here, right? And so the double-strand break pathway often results in crossovers. Okay, and this is the model of how crossover of homologous recombination occurs during meiosis. And we'll talk specifically about crossing over during meiosis in a few slides after we've explained these models. So, for example 1, all right, back to this picture again, whether recombination in the holiday junction pathway in that model results in chromosomal crossover or not is determined by how the double holiday junction gets resolved. So, chromosomal crossover will occur if one holiday junction is caught on the crossing strand and the other on the non-crossing strand. So what that means is, if we look down here, right, so this is the double-strand break repair side, okay? So if we cut at the purple here and the purple here, all right, we will get a non-crossover product. Okay. If we cut at the orange arrows here and the orange arrows here, we will also not get any type of crossover. But if we cut at the orange arrow at this first holiday junction and the purple arrow at the second one, we'll get a crossover. And vice versa, same thing. If we cut at the purple arrow here and the orange arrow here, we will get a crossover. So if we cut at both purples or both oranges, no crossover. If we cut at one orange, one purple, we will have a crossover. Okay, and having a crossover is more common than having a non-crossover product. All right. So let's draw this out so we can see what's going on. We'll use different colors so we can see the different chromosomes. All right, so first things first, we have a break in the chromosome. All right. So what I'm going to do to keep track of what our DNA has is I'm going to label 3 different loci, okay? And that's just a piece of a region of DNA. So I'm going to say that this is region A, okay, locus A, locus B, and locus D. All right. And now the homologous chromosome is going to be unbroken. Okay, and we're going to label these with little a's so as to differentiate them. Okay, all lowercase. So first things first, after that break, we go through resection. Okay, now we're not going to mess with the homologous chromosome, okay, because there's nothing wrong with that one. Let's see. Okay, so what we've had here is resection 5' to 3'. Right? So that leaves us with some overhangs, right? So some 3' overhangs. Now, we can go through strand invasion. All right, so let's continue on down here. Okay, so what we have our 3'. We have our 5'. Okay, we have that A. We've been resected. We have B and D. All right, we have A. And what we are going to do, let's draw out this one. Okay. So what happens here is that this strand starts to invade and pop up. Okay, it pops up this top 5' strand. Okay, up here. Okay, it moves this up. All right, and this right here, this dotted line, that's all new synthesis that's going to be occurring, right? And this is the strand invasion process happening. All right, and so we're going to start synthesizing this way, right? Okay. Now, what we are going to be doing, since we're synthesizing using this bottom strand as a template, remember the top strand up here, right? It was big B, right? Well, what we're doing is, since we're using this as a template, this is actually being made as a little b, okay, as that locus. So it's matching the homologous chromosome, not the original chromosome. And for example, let's just say what this big B versus little b could be. So let's say that big B that we're seeing, let's say that that is an AT base pair. And let's say that little b is a GC base pair. Okay, so that might make it more easily understandable. Okay, so there is the start of our strand invasion. Now, what we can proceed with is the full double holiday model. Okay, so double strand break repair. And this is what we are going to have. Okay, so now we have the full, oh, and what's happening here is that this is being synthesized using this piece of red as the template. Okay. All right, so what we can do is if we call the crossing points, right? If we call these points, this is where we're going to have to resect them. So if we say that this, right, you're either going to cut here, let's do it in different colors so we don't get confused. So you can resolve it by cleaving there, right? Okay, in those places. So here is what we can do. Okay, so if we decide that we want, so what we're going to do is go through resolution. Okay, so if we resolve this, okay, so we separate it. If we resolve it at both green and green or purple and purple, this is what we will get. Okay. So this is at both green or both purple arrows. Okay? Now, if we do this at one green, one purple, okay, so let's say the first one green, the second one purple, or vice versa, the first one purple, second one green, this is what we'll get. Okay, so these right here are considered non-crossover. These down here are considered crossover. Now, why? Okay, first of all, what we're looking at is A, locus A with respect to D. Okay, the original molecule, okay, big A is found with big D. Little a found with little d. Okay, so that's what we consider as normal or wild type. So after all of the finishing of this, right, the resolution, we see that A is still on the same chromosome as big A with big D and little a with little d. Okay, so we're not looking at the middle right now. Okay, so that is, we don't have a crossover. Now down here, we see that big A is now with little d, and little a is with big D. So A with respect to D, there has been a crossover. All right? Now, before I mention something that you might have already looked at, okay, right here, we're going to keep this for one more second, okay? Okay, as you can see, we have big B, little b. Okay, but first, how did I get these? How did I get that from this? Okay, well, let's show you. Three prime, right? A, B, D. 5 prime. Oh, that should be an A. Okay, so we'll make this. Ooh. Okay, so let's show you how we get this. Now, the easiest way to look at it is, what if I cut it at both purples? Okay, so what that's basically saying is, what I'm going to do is, if I cut right here, it's basically like me erasing, connecting these right here, connecting that right there, erase this, connect it right there, connect it right there. So what we're seeing is this being A, B, D, big, and then A, little b, big D. Okay, so that's what's happening over there. What's happened down here? Okay, the bottom strand isn't touched at all, right? Just like the top strand wasn't touched. So we have little a, little b, little d, and then what do we have? We follow it. Okay, we can just follow it. A, follow up, little b, follow up, down, little d. Okay, so that's the same thing we get over there. Good. Now, what if we cut Okay, what if we cut at both the greens, right? So what does that look like? Well, what we can think of is we're not touching the inner strands at all. And what we're doing is when we make the cuts here, we're cutting both strands. Okay, so cut there, cut there, cut there, cut there. And we're just making a big X. So this down to the bottom and up to the top. This one up to the top, all the way over, down to the bottom. So all you have to do is follow along. So what we can see is we have big A, okay, big A, down to little b, up to big D. Okay, and then the other strand is big A, little b, okay, no movement, big D. Okay, over here, little a, little b, little d, no change. Up here, big A, or sorry, little a, big B, down to little d. So A with respect to D, we still have no change. Okay, you might get a little difference between the locus in the middle at B, but there's no change A with respect to D because big A is still with big D, little a still with little d. Okay, but when we cross, okay, when we do one of each, we're going to see something different. Okay. Okay, so now let's say we cut here and here. Okay, so what's that going to look like? Once again, it's just like you erased. You complete those. One here, right? We've cut here. So you just make the big X. And what does it look like? So, top strand, okay, big A, big B, okay, we'll follow it in black. Big A, big B, down to little d. Okay, so we can already see that A with respect to D is different. Okay, this next one, we have big A, little b, little d. So A with respect to D, crossover, right? Down here, little a, little b, up to, all the way up, big D. So A with respect to D, crossover. Down here, little a, little b, all the way up to big D. And sorry, the previous strand, this is little a, little b, still big D, but it's the bottom one of the big D's. Okay, so on both strands, A with respect to D has had a crossover. So we're looking down here. Okay. Now, the last thing that I pointed out, and I said, let's take a look at this. With each of these, we can have what's called a heteroduplex. So that is when we have, let's say, on this strand, right, it's big B, so maybe it's a T up here. And this is a C, okay, just for example. Okay, so that has to be repaired. So if the heteroduplex is repaired by mismatch repair, back to the original, let's say in this case, it's supposed to be big B, then everything's okay. But, okay, see down here, so if it's big B, everything's okay. Okay, big B up here, everything's okay. But down here, what if I say that this is repaired the opposite way, right? So let's say over here, this is repaired to little b. Okay, well, that's what we call gene conversion, okay? Because it was supposed to be big B, so it should have been an AT, but now it's a GC bond, okay? So that's going to lead to what we call LOH, or loss of heterozygosity, because remember, right here, okay, originally it was an AT pair. And on this chromosome was a GC pair. So, GC, okay? So that you have heterozygosity at the B allele. But if they're both repaired back to an AT or a GC, you lose the heterozygosity, you're homozygous at that allele, and that could be problematic. So it can lead to disease. For example, this can occur when you have a loss of heterozygosity at a certain locus. That can lead to something called retinoblastoma, which is a type of cancer that affects the eye, okay, the retina. And this can be highly problematic. All right, so we talked about the Holliday junction model, and now let's say the other type of double-strand break repair. Okay, so we have synthesis-dependent strand annealing, which is SDSA. Okay, so homologous recombination via the SDSA pathway occurs in cells that divide through mitosis and meiosis. And this will always result in non-crossover products, so repaired back to as if nothing ever happened. Whereas homologous recombination via the double Holliday junction model more often results in crossover products. So let's talk this through. So we have the Holliday junction being formed, okay, just as if it were the other way as well, but instead it's resolved via branch migration. Now, the invading 3' strand gets extended along the homologous, right, the recipient DNA duplex, and this is done via DNA polymerase. And it gets released as the Holliday junction slides, okay? So we call it branch migration. It's sliding, it's moving. So the newly synthesized 3' end of the invading strand, remember, it's that nucleoprotein filament, nucleic acid proteins, okay? That can then anneal, okay, so base pair, to the other 3' overhang in the damaged chromosome through complementary base pairing. And then after annealing, we can cleave off some small flaps of DNA. However, this is not resolved, okay, the Holliday junction isn't resolved via cleavage like the Holliday junction model is. This one is resolved by moving and basically pulling back up the strand. And I'll draw that out for you in the next slide. Okay, so here's an example. Let's show our SDSA. So here's our 3'. All right, we still have our A, we have our break. Here's our B and D, B and D, A, A, B, D. And by the way, on both of these, I have just said that there's a double strand break here, and anything that's missing there, If it's a part of a gene, that's going to break that gene. If you have any part of a missing gene, you're going to have problems down the line in transcription and translation. As I've written it here, I'm saying that it's not an important piece of DNA. Okay, but that's not always the case. All right, so just like the holiday junction model, after double strand break, there's still the resection. Okay, in the remember this is SDSA. So we have resection. And we have 3'. And remember right here, nothing's happening on the homologous chromosome. Okay. Now, we once again have strand invasion. Remember, we always want to get our polarity right so we know which way we're going to be synthesizing. Okay, so there's strand invasion. Now we go through a process of branch migration. Okay, and what is happening there is what we're seeing. Let's draw this out. Okay, and this is being synthesized, right? Let's not forget that. Okay, so this is what it's doing is this is moving in that direction. Okay, so the holiday junctions are just moving, and then what we end up with is instead of cleavage at this spot, okay, or these spots, we have what's called dissolution. Okay, so no cleavage. And what we're basically doing is we're pulling, say, this blue strand, we're just pulling it up. Okay, we're pulling it up back to its own chromosome, and we're pulling this red one back down to its proper complementary strand. So what we end up having is okay, if we follow it, we have our A, our B, and our D, right? We're just following all the way across this line. And this one, since we're pulling up, okay, we're going to have big A, little b, big D, because we're just going to pull that back up. Okay, so A with respect to D, no crossover. Now let's look at the homologous chromosome to check it. Okay, we have the bottom one's the easy one. Okay, little a, little b, little d. It didn't have to worry about anything. And then this one, remember, we're just going to pull it down. So little a, little b, little d. There's not going to be any cleavage. So these are a non-crossover. However, let's still look. We do, in this the originally damaged strand, we have a heteroduplex. Okay, so this heteroduplex still has to be repaired via MMR. Okay, and if it's repaired back to BB, it's like nothing ever happened. If it's repaired back to, or repaired to little b, we have gene conversion and loss of heterozygosity. Okay, so remember, instead of cleavage, SDSA goes through dissolution, and that's where we have the 5' blue strand just pulling back up and becoming the template for the 3' blue strand. So it can be, we do the synthesis here, right? And then it comes back up, and this synthesis, right, is just coming back off of the blue strand, not on the red strand as seen in the homologous recombination via double holiday junction. Okay, and so once again, we can still have that heteroduplex, and that can be affected whether we have gene conversion or not based on how mismatch repair repairs that. All right. Great. Okay. Now those are the big boys when we're talking about homologous recombination, but there are other types of homologous recombination. So we're going to go over a few of them, starting with single-strand annealing. So the SSA pathway repairs double-strand breaks between 2 repeat sequences, and this is important. Okay, the sequences have to be repeats. So this doesn't require any type of homology between 2 different homologous chromosomes or identical chromosomes. Okay, so this is using just the single double-stranded helix. All right, so this is unlike the holiday junction or SDSA pathways. This only requires that single DNA duplex, and it uses repeat sequences for the repair. And those repeat sequences need to be at least 300 base pairs, or sorry, 30 base pairs long. Okay. Now this is considered extremely mutagenic because it results in large deletions of DNA as you are searching for the repeats. All right. And I will show you an example of this in just a couple slides once I finish talking about SSA. Now, as DNA, if we're going to talk about specifics, as DNA around the double-stranded break gets resected, the single-stranded 3' overhangs are coated with the RPA protein. Remember, RPA is going to help protect this from endonucleases, as well as help form that nucleoprotein filament that can go through strand invasion. Right? So RAD52, another protein, would then bind each of these repeat sequences on either side of the break and align them so that they can anneal. After they anneal, the leftover non-homologous flaps of these 3' overhangs get cut away by endonucleases, And then new DNA synthesis will fill in any gaps, and DNA ligase will ligate those gaps, okay, making them continuous strands. So the DNA between the repeats that bring off as flaps are always lost, as is one of the two repeats. So this is highly mutagenic because you lose a lot of sequence. So let's see what that will look like, right? So we have our 5' to 3' resection, our homology search, our annealing of those overhangs, the cleavage of the flaps, and then synthesis and ligation to finish it. So let's see. What's this look like? So here's our strand. These are regions of homology. Okay. Now, we get a double strand break. Okay. Okay, those are the repeats. Okay. So first things first, we undergo resection to find that homology, right? So we're going to resect. Okay, we'll continue on this line. 5' to 3' resection. Okay, so it's basically like your eraser. Okay, and let's make sure we still know that we have the repeats right here. Okay, so the resection, right, went in a 5' to 3' manner, so it's basically just doing this, right? It's an eraser function. Okay, good. I'll draw this back. Okay, all right. Now, the next step will be single strand annealing. So we find that we do the homology search, right? So we're looking for these red spots, and then we do the annealing of those overhangs. So what it looks like is this. Oop. And this is going to be single strand annealing. Okay. And what it looks like is this. Okay, so what you're doing is you're just moving these close in that direction, right? So you're pushing them together until these repeat sequences overlap. So this right here would be the two of them, right? And so you're going to lose one of them so that you can actually make, okay, so you'll lose, let's say, the bottom half of this piece and the top half of that piece to make one together. Okay, so there's your single strand annealing. Okay, so the next step, we're going to go through cleavage. We have to cleave those flaps. Okay, so what we're doing, okay, so what we've done here is we've nicked. Okay, we nick it. Okay, to cut it off. All right, and then finally, we go through the DNA synthesis and ligation to close the gaps. Okay, so here's that one repeat. We've lost one of the repeats, right? As well as, let's say for example, so what we've ended up losing, if we compare this to the original, we've lost everything from here to here. So we've lost a lot of sequence, right? That all gets lost. Now, it's better to lose that and it be somewhat mutagenic than for you to have to completely lose this piece of chromosome. However, not our preferred mechanism of repair. Okay. Now, another form of homologous recombination is called break-induced replication, or BIR. And this pathway repairs double-strand breaks encountered at replication forks. And this is because DNA helicase is trying to unwind the template strand, and it leads to, or it finds a double-strand break. So the exact mechanisms of BIR aren't exactly clear. There are several proposed mechanisms. 3 of them all have strand invasion as the first step, but then they differ in how the D loop migrates as well as some of the subsequent events. But we do know that BIR pathway is actually very useful in maintaining the length of telomeres when telomerase, the enzyme, is either inactive or not present in certain cells. All right, so when we talk about meiotic recombination, remember, homologous recombination is what's occurring. Okay, so in meiosis, homologous recombination is required for proper chromosome alignment and segregation. So in meiosis, double holiday junctions always get resolved as crossovers. So if we think about our homologous chromosomes during meiosis, right, we have homologous chromosome, right? And when they come together, they'll form what's called a chiasma. Okay, so right here, this is called a chiasma. And this helps a cell go through proper chromosomal segregation. Without chiasma, you are actually increasing by far your occurrence of what's called non-disjunction. And that is when you have, let's say, both homologous chromosomes ending up in the same sperm or egg cell, and then one egg cell being without that that complete set of genes. So let's say, for example, one sperm would get 2 chromosome 21s, right? Whereas another cell will get 0 of them, right? Well, if this chromosome 21 sperm cell fertilizes the egg cell that was produced normally, so it has 1 copy of every gene or 1 copy of every chromosome, but this sperm has one copy of every chromosome, but two of chromosome 21, right? If these guys fuse, you're going to produce a fertilized egg with three. It's a diploid cell, but instead of 2 chromosome 21s, it has 3 chromosome 21s. This is a disease that we call trisomy 21 that may lead to Down syndrome. That leads to Down syndrome, okay? So chiasma are very important. So in a chiasma, we are undergoing crossing over. So we're crossing over genetic material. So this black one, when we resolve this, what this looks like is that we have a little bit of the red, okay? And this one has a little bit of the black. So not only does it help us with non-disjunction events, but we actually have some gene transfer, which helps with the variability of the genetic sequence and can help with evolution. Okay, so this will always, how we resolve this in meiotic recombination, this will always result in a crossover product. Okay, good. So in meiotic recombination, we can talk a little bit of the detail. We have this protein called SPO11. All right, it is going to make a double strand break at what's called a recombination hotspot. So it's a place where it is likely to occur. All right, now we then resect. All right, so we cut away by we're using our mRNA complex. Okay, so so here's SPO11 occurring. Here's the resection. Okay, then we have RAD51 and DMC1. These are proteins that coat the single-stranded DNA and help with the strand invasion. So these are involved in making the nucleoprotein filament and making the holiday junction. So this is occurring in here. From there, the holiday junction migration can result in heteroduplex DNA containing mismatches. So we're coming down here. Okay. Now the gene conversion can result in heteroduplexes like we talked about before. So we can have a loss of heterozygosity. Okay. So instead of, big B on one strand, little b on one strand, maybe we have both of them being resolved to all little b's, right? So you'll have a all one way versus none, the second way, right? So we're resolving our crossovers. So we've already talked about how they get resolved. So that was just a quick overview of just another few specific proteins involved in the meiotic recombination. Okay. We've only talked about eukaryotes so far, so we have to talk just a little bit about double-strand break repair in prokaryotes, and their pathway is called the Rec-BCD pathway. And there's basically just the one pathway that they go through. And what happens is that we have this Rec-BCD protein complex binding to and unwinding our double-stranded DNA end, okay? And that results right here, so they're binding here. They unwind, creating single-stranded ends, okay? As well as a single-stranded loop. Okay, the two tails will then anneal, reanneal, okay? To produce our second loop. Okay, so this is loop 1, this is loop 2. Okay, now both loops can move and get bigger, all right? What we then have is Rec A is going to be binding, right? It's going to be binding to the DNA to protect the single strand, right? We don't want it to get nicked by endonucleases and then fly away. And then we have Rec-BCD coming in, okay, adding on, right? It's already there, but it's now we're going to actually cause a nick, right? So we're going to nick this 3' strand at what's called the chi sequence. And this is just a hot spot for where we have nicks from the RecBCD complex. Okay. We then unwind further. Okay, and then we have what we see as this really long, okay, 3' end with the chi sequence near its end. So this chi sequence right here, okay, if we follow it all the way back, here's the chi sequence. That's just the hot spot of where we're going to nick. Okay. Now, if we continue on to the next slide, we've already shown that RecBCD loads Rec-A. Okay, so that's down here. And once Rec-A is loaded, RecBCD can disassemble. So it comes off of the DNA. Now, the Rec-A promotes the strand invasion, okay, into the homologous DNA duplex, producing our D loop, the displacement loop. And then the D loop gets cut and anneals with our gap in the DNA. So this is happening right here. And then we can resolve this DNA, this complex, right? This holiday junction complex via the RUV-AB complex. Okay, that's going to bind, right? The RUV-AB is going to see this holiday junction and come and bind. That's then going to recruit RUV-C, and we are going to resolve it. Okay, so this is all occurring here as well. And this is our final product. Okay, so that's how prokaryotes would do this, or E. coli specifically. So we did all the details in eukaryotes, then we kind of zoomed over the prokaryotes a little bit. But this is the overview of how prokaryotic homologous recombination repair occurs. Okay, so to compare our prokaryotic and eukaryotic recombination, the pairing of our homologous DNA, that's Rec-A in E. coli and Rad51 and DCM1 in eukaryotes, DCM1 specifically in meiosis. When we're generating that single-stranded DNA for strand invasion, all right, that's RecBCD in E. coli, or the MRX complex in eukaryotes, MRX is yeast, MRN human. And then holiday junction, recognition, branch migration, and resolution, that's the RUV-AB complex and RUV-C for resolution for E. coli. And in eukaryotes, we have a much more complex set of proteins handling that. All right. So that's all of our homologous recombination. All right. There is a different type of recombination, and instead of doing a whole long unit on that, I just added a few slides in this unit just to keep it all in recombination. So we have what's called site-specific recombination, and there are two types that we're going to talk about. One is called conservative site-specific recombination. All right, and that is involving protein enzymes called recombinases, and they act really similar to topoisomerases. Okay, so they cleave and rejoin DNA molecules to either invert DNA fragments, okay, so to turn it upside down, okay, and flip the sequence of events, or to insert them into a new site, okay, so you either take it from one site and move it to the other. Okay. We also have what's called transposition. Okay. Now, this uses a different type of enzyme called a transposase. And this transposase enzyme will cleave two ends of a transposable element, and it'll randomly insert it into another place on DNA. Okay, so which we call the target site. So, what are these going to look like? So let's talk about our transposons first. Okay, so transposases will cleave both ends of the transposon in the original site and catalyze this integration into a random target site. So we saw that on the previous slide. They can do this in what I like to call a cut and paste mode or a copy and paste mode. And the difference between that is such. Okay, so Here's double-stranded DNA. Here is our transposon or transposable element. Okay. Now, we have two options. We have the cut and paste option. Okay. So that is this. That is where we remove it from here. Okay, so it was here. We've cut it out of the DNA, and now we've moved it to another spot on DNA, which is right here. Okay, that is what I call the cut and paste version. We can also have the copy and paste, which, as you can probably already imagine, we leave the original piece of DNA, right? Sometimes we call these jumping genes, right? Transposons. We leave the original one and we copy it and add a second one in a new site, all right? So we started with one. In the cut and paste, we still only have one. In the copy and paste, now we have two. Now, as we see down here, more than 40% of the entire human genome is composed of repeated sequences. And it is very likely that a transposon got into our ancient ancestors, and throughout evolution, okay, throughout the time, it has not only cut and pasted itself, but more often, more likely, copied and pasted itself, making those repeats show up in many different parts of our genome, meaning that we've also lengthened our genome as we've grown as well. So the transposons are very likely responsible for a large portion of these repeated sequence. Okay, and we have a lot of repeated sequence in our genome. Okay, so I want to give you an example of our site-specific recombinases. Okay, so here's an example. All right, what if we have a circular piece of DNA and it wants to recombine, okay, so plus, with a linear piece of DNA. So this can sometimes happen, let's say, when a bacterial plasmid, right, a vector, is going to join into the main genome. Okay, so what is important here is that we have to see these recombinases work with a polarity. So this arrow that I'm showing isn't just for no reason. This means that they have the same polarity. So what you can do is to insert into this, A will cross like this. Okay, we do a cross like this. And what we end up with, we just follow the polarity. This is actually pretty easy. So we can start at X, okay? We just start at X, okay? We have a rightward arrow, and then we follow it up, okay? It goes to B, comes all the way around, okay? DNA sequence to A, and then A crosses back down, okay, rightward arrow, and then we get to Y. So this could be one like nice long linear sequence from a circular sequence and a small linear sequence, okay? And this is what we call insertion. Okay, so we've inserted this piece of DNA, okay, the circular piece, into this linear piece of DNA. Okay, there's also what's called inversion. And what we would have here is, if we start with X, rightward arrow to A, long piece of DNA, B, and then we have a different arrow, a leftward arrow to Y. Well, these polarities have to match up. So what has to happen to this is we actually have to curve this around, okay? Right? Because look, the arrow was pointing at B, so it's always going to be pointing at B, right? And so how we do this, we're going to once again make a nice X. Okay? We can start with X, right? So we're going to go this way. Okay? So what we have, we start with X. We have a rightward arrow. Okay, we come to B, follow it all the way around to A. Okay, so a long piece of DNA to A. Then what do we have? We have an arrow that's going to be pointing at A. So a leftward pointing arrow. And then down to Y. Okay, and this is what we call inversion. So this is when we can flip a DNA sequence, okay, as we see here. Look what's happened. We still have X and Y on the outside, but in this case, A to B went from left to right. Now A to B goes right to left. Okay? Now the last one that we're going to show is a deletion. So how can we take something out of the genome? All right? So if we see here, we have X, rightward arrow to B, nice stretch of DNA, A, rightward arrow to Y. Okay. Well, what we can do is we can circle that up. Okay, X to B. A to Y. Okay, we can make our nice red cross again. And we can draw it out. So, coming back down here. Okay, what do we have? We're going to go X down to Y, and that's the end of the DNA. So we're going to have a plus. All right, and then what about this one? We can say this is A going to B, and it's circular. Okay, so this is A going to B, and it's circling around back to A. So this is A almost back to up here, right? So we've started with this again. Okay, this is called deletion. So this is how you could insert something in. Okay, so you can insert a piece of DNA into a host cell genome. Okay, think of viruses do this. You can invert something, a piece of gene. Okay, or you can cut yourself out of the genome. So think of our viruses, let's say, that jump into the human gene and lay dormant for a while. Okay, so they've inserted in there, and now when they want to be active again, they can cut themselves out, say deletion, and become active again. So these are some examples of our site-specific recombination. I hope you enjoyed this lesson, and I hope to see you back. Thank you very much for joining us at educator.com.